Today is Monday, April 4th, 2016. I am Bryan Mackenzie. I'm here in the Senior Physiologist Lounge in the San Diego Convention Center during the annual meeting of the American Physiological Society. Today I have the pleasure and honor of interviewing Dr. Marcia Linder for the Society's Living History Project. Dr. Linder has been affiliated with California State University at Fullerton since 1977. Her research focuses on the transport, storage, and metabolism of copper and iron in mammals and how these are perturbed in gestation, lactation, and disease states. Marcia, welcome to the Living History program, and thank you for agreeing to be interviewed.
My pleasure.
If you're ready, I'd like to ask you about your career. Can you tell us about your family upbringing and how you became interested in science?
That's a very good question. My father was a physician, and he had friends who were scientists, including the first biochemist I became aware of, who was a family friend. But also the school I went to, which is a Waldorf school, so-called Waldorf school, they're all over the world. They try to expose all the students to all kinds of things, from all the arts to all the sciences. And they start in grade school with phenomena, exposing you in terms of science to phenomena rather than dogma. So it sort of stimulates you, I think, as a child to be interested in, hey, what's going on here, this kind of thing. So I always had an interest in how things work, and my parents were very much oriented towards looking at nature and looking at new things, observing. I guess I was sort of taught by my family to observe. And then, so I really wondered whether I should go into medicine, in part, I think because of my father, but also I was very interested in all kinds of science. And then during college, I did a summer research project with this biochemist, and it was working on the DDT, I had to determine chlorinated hydrocarbons in the environment. This mentor biochemist, Dr. Ehrenfried Pfeiffer, was also very instrumental in the beginnings of the organic agriculture movement. Mm-hmm. So he was trying to find alternative ways to raise crops that did not involve the use of pesticides and herbicides, and that interested me as well. I've always been interested in things that are not necessarily in fashion, put it that way. So between all of those things, that's how I sort of got into it.
Okay. So you received your BA in chemistry from Vassar and your PhD in biochemistry from Harvard. Can you tell us about your academic training and how you became interested in pursuing your career track?
Well, when I got to, one of the things that interested me was that I went to the medical sciences program at Harvard, which was a new NIH-funded endeavor to try to expose graduate students, PhD students, to not just your particular science, I was going into biochemistry, but also to physiology, anatomy, even histology, psychiatry, all those things. And so we had some of our own courses, but we also mingled with the medical students, and I think that was interesting in terms of, and then I had to decide who to go with in terms of my mentor, and I was interested in brain function, thinking that might be interesting, and regulation in general, how things are regulated metabolically. And I ended up with Gene Knox, who had discovered the enzymatic basis for amino acid deficiency diseases. I should say enzymes, amino acid metabolism enzymes, with phenylketonuria, for example. I didn't like what was happening in brain at the time, which was just analyzing the kinds of fats that were in the brain. And that didn't interest me nearly as much. So that started me on this. Gene Knox was the first person also to discover substrate-type regulation of enzymes in mammals. And the mechanism is very different from that in bacteria. And at the time, Jacob and Monod were dominant in the field, and regulation was always by repression of gene expression. And here in mammals, it was the opposite. There was a completely different mechanism, and that really interested me. So I got involved in sort of substrate-type regulation, which led to my going into ferritin, because iron was regulating ferritin, and that was over at MIT, so I joined Hamish Monroe there and did a lot of stuff still on iron even today as a result.
So tell us a little bit more about Hamish. You worked as a postdoc at Harvard and then at MIT. Tell us about those experiences.
Well, during my PhD training, I was working on the regulation of tyrosine aminotransferase and tryptophan oxygenase. But my mentor got very ill. So I didn't perhaps have as much mentoring as I should have had. Although I did a lot of research and I thought I discovered some interesting things. But so he put me on a different project which had to do with cancer. He had the theory that there were certain key enzymes that all cancers turned on. And he was interested in my looking at one of them, which is glutaminase, because glutamine is a big nutrient for cancers. And so I got into the cancer field that way, so that got sort of interesting. So when I went to MIT then to work on ferritin, I explored whether there was anything interesting relating to cancer in iron metabolism. And it didn't look all that fascinating at the time. So I started looking at copper, and there was all this information that blood copper levels were increased in cancer. So we decided to look into what it was that was increasing. And I was still working on ferritin at the same time with Hamish Monroe, and we continued to collaborate until I left MIT, which was throughout my time as a professor at MIT.
So after you were on the faculty at MIT, you moved to California State University, Fullerton in 1977, and there you served as chair of the Department of Chemistry and Biochemistry from 2004 to 2010. Of course, Cal State Fullerton and MIT are very different institutions. Tell us about that transition and your experiences at Cal State.
Yes, well, MIT, coming from Harvard in general, at least in those days, had an atmosphere of being more, people there were collegial and culturally oriented. We went to our twice a month concerts with the Boston Symphony and things like that. We went to museums. When I got to MIT, it was a very different atmosphere. People didn't want to talk with each other. It was as if they were just focused on their work and they didn't want to share their ideas because they were afraid that they might get scooped. So the students were wonderful, and they really made it for me at MIT, but there were very few women faculty, and there were a lot of professors who were not very friendly to women. That didn't necessarily worry me, but it was interesting being in an atmosphere where there's a lot of, it was just at the beginning of the women's movement too. So when I was first an assistant and associate professor at MIT, and I think there were 7 of us women faculty at that time out of about 700. And it was interesting, but I wasn't sure I wanted to stay there the rest of my life. And then I met my husband, which is why I went out to California and looked for a position. There weren't many openings, and I was really impressed by my colleagues at Cal State Fullerton in the Department of Chemistry. At that time, there was no biochemistry attached to it. There were 2 other biochemists. I was the third to come there. And my 2 other biochemistry colleagues and also the other chemistry faculty were very impressive. They were real scholars. They really loved ideas, but they also loved to teach. And I also enjoyed interacting with students. And I felt I could do my research there and also enjoy my teaching and mentoring of students, even though we didn't have a PhD program, just a master's program. And it's turned out to be extremely rewarding. You know, it's sort of you switch from being a small fish in a big tank to a big fish in a small tank. They both have their problems, pros and cons. And I think probably it did inhibit my getting some more grant funding going to a non-PhD granting institution. But there were a lot of wonderful aspects to it, which is why I'm still at it, even though I'm pretty old.
So you mentioned that you were one of 7 women on the faculty among 700 or so at MIT. Early in your career, who were the women in science who inspired you?
Actually, I didn't have a lot of female role models in the sciences. I did have one who was a family friend. She worked with this biochemist I was telling you about, and she was a PhD. And she certainly was a role model of sorts, but I had very good men mentors, actually. I was lucky. Also at Harvard and then at MIT. Both Hamish Monroe and Jean Knox enjoyed working with women, I think more than men. And they were very helpful, really. So I've been lucky.
You described your foray into ferritin. How did your interest in metals develop, and who influenced you in that path?
Well, working with Hamish and iron on iron got me very interested, and starting to teach eventually too. What started me, well, one of the things that made a difference was that I was involved in a project with my students to put together all that we knew about iron and in terms of nutritional biochemistry. And that, you know, when you start looking into all that is already known, that stimulates your ideas about things. And then the copper area got very interesting, you know. So it was, and copper and iron have interactions. And so there are so many questions that come up when you get in, start to study what's already known. You know, I think scholarship is really important. Reading what has been done. It's getting very difficult these days because there's so many people working in every field. It's hard to keep the big picture entirely intact, but if you've been in it for a long time, it helps at this point.
So looking back, do you think you can identify what might be your most important research contribution?
Yeah, I don't know. There's a lot of them, and I don't think I can point to one. I really can't.
Well, tell us about 2 or 3.
Well, I don't know whether I should start from the beginning or more recently. I'm really excited about some of the things I'm doing right now, for example. I think we're into finding a new copper component in the blood plasma, which might be very interesting, and in terms of understanding the homeostasis of copper in the mammalian organism. Svetlana Vitsenko's group found a small copper carrier in the urine of Wilson disease model mice. And we're collaborating with her on this project. And one of the things that we discovered immediately was that there is a lot of this in the blood, as you would expect. If it goes into the urine, it's either coming from the blood or it's being secreted by the kidneys. And we have high levels of small copper carriers also in the blood plasma. And these carriers increase in response to this liver loading of copper that occurs in the Wilson disease mice. But what happens is there's an adaptation, which the adaptation is that small copper carriers go up in the blood and go out in the urine, and liver copper levels go down. So this might be an alternative means of excreting copper, and we've, the dogma has been, and of course there's good evidence for it, that the main way copper is excreted from mammals is through the bile. The dog also has a lot of problems with copper toxicosis. It's sort of unique in several ways in terms of how it handles copper. It tends to have, even normally, at least 10 or 20 times higher levels of copper in the liver than other mammals. It has an albumin which doesn't bind copper with high affinity. And now we found this small copper carrier, which in a lot of dogs seems to be elevated in the blood plasma and urine. So some dogs may be producing a lot of this small copper carrier, which allows them maybe to survive better. So this is something that we're exploring now, and we have collaborations also with veterinarians on this, Hille Fieten in the Netherlands, for example. So I'm really excited about that project. One of the big difficulties, of course, is to characterize this small copper carrier and get enough of it for solution NMR. And to get funding for something like this, you really have to get that before you can. So it's like a catch-22. But that's something I'm really excited about. So we also, let's see, in terms of contributions, of course, I have been one of the people studying, my lab has been studying the functions of various copper proteins in the blood plasma particularly. And what we had evidence for many years ago was that ceruloplasmin, which is the most multifunctional protein you could possibly imagine. I'm just writing a review about it which is pointing out just how many things we're still discovering about all the things it's doing. And it's the main copper binding protein in the blood plasma, and the copper there doesn't come on and off easily. It gets buried in the structure. So people were very averse to the idea that the ceruloplasmin copper might actually be entering cells by interaction between ceruloplasmin and cells. We've just published an article where we think we put that question to rest very clearly, that it is a copper transport protein. It does interact with cells. So I finally gotten it all together to show that, something I've been wanting to do for years. So there's a couple of ideas.
Thank you. You mentioned that you are writing a review on ceruloplasmin. You've also been the author of 2 books, The Biochemistry of Copper in 1991, and the textbook and reference volume, Nutritional Biochemistry and Metabolism in 85 and 91. How was it for you writing and editing those books, and what do you see as the impact of those publications?
Actually, those are some of the things I'm almost the most proud of in a way. I loved to write already, and as an undergraduate, I took all the writing courses I could take at Vassar, and I had equal numbers of credits in the liberal arts as in the sciences. So writing has always been easy for me, which is an advantage, I think. But I love to try to put ideas together, put it all together. And so I got into nutrition. I started some nutrition courses. Actually, they were started before I got there in the chemistry department. It's a way of teaching science to majors in other disciplines, non-science majors. But the textbook that I wrote, or handbook that I wrote in nutritional biochemistry, it was wonderful to learn about all the different aspects of metabolism that I had to do to write it. And it's still, what I put in there was, I looked up the research behind it, and it's still pretty much valid. I'm very pleased about that. In fact, it does a pretty good business on Amazon still. The last edition was in '91. And the same is true for my Biochemistry of Copper book. It's still being sold. Every year I get royalties still. Not very large, of course, but it's amazing to me. So, one of the things is that if you are able to put together the data that makes sense, then in a way that they make sense, that's lasting. That's a lasting thing, and other people can benefit from it. So I'm pleased about my contributions there. I might also add one other thing. I've had this ecological interest, and I told you about organic agriculture. I think actually my book influenced nutritional policy because I put in information about what kinds of trace elements and vitamins we lose when we process whole grains. Typically, even today we're still eating mainly white bread and white rice, but there's so much more awareness now of what happens with that and the loss of fiber and all these trace minerals and vitamins. So by putting together the data and putting it in this nutrition book, I think it helped to make people more aware.
Great. What role do you think scientific associations have played in your career?
Well, one of the great things is, of course, these conferences. It gives you an opportunity to interact with your colleagues, and APS and ASN have been particularly good at also, over the decades, of bringing together a lot of people in my area of interest, iron and copper, attracting the researchers in the field. So unfortunately, it's not quite as active now as it used to be. But things change, and of course there are all these other conferences that involve trace elements. So, and also, they're willing to put on special sessions if you make the effort. I haven't been as involved in, these societies themselves and contributing to running things as I might have been, but there's so many other things to do. You can't do everything.
So what was your first scientific meeting that you attended? How was that for you, and how did it differ from scientific meetings today?
It was it was then, in those days it was called FASEB, the FASEB meetings, and the first ones were usually they were in what's that place called in New Jersey?
Atlantic City?
Atlantic City, that's right. Actually, my very first one was in Chicago. And I was alone. My mentor didn't come with me. I think it's better if mentors come with their students. But I had a good time, and I had to give a presentation, and nobody was there, and there were like 500 people in the room. And it was interesting, and I felt I did a good job, but it was, so I was not, people today are mentored much more than I was in those days. It has some of the same atmosphere in terms of just many, many people in big sessions. Yeah.
So you mentioned that at Cal State Fullerton, you have no PhD program, and as I understand, your lab is populated mostly by undergraduate students and master students. Mm-hmm. Tell us about the challenges and the satisfactions of working with a younger, fresher group of students.
Yeah, when you're trying to bring students up to snuff so they can actually produce reliable, reproducible results. They usually, don't end up doing it right away, so you waste supply money on them. But, overall, it's amazing to me, still today, how well they do. They really can do really good work. And in our department, we've had a research focus ever since the university began. We had scientists that were presidents and chemists, especially. And so one of the things that came out of that was that in chemistry and biochemistry, all our degrees require research. So all our students spend at least a year doing research, and a summer usually, but most of them do 2 years or so. So you end up having them in your lab much longer, so they can actually contribute much more, and some of them are real stars. We have a lot of disadvantaged students, under-resourced students, not just ethnic minority students, but also disadvantaged from financial and educational points of view, and they're real wonderful students in there. They're really great. And, seeing them achieve something and is really rewarding.
So where have those students gone on to and discover
Well,
success?
one of the things that we do in our department particularly, because, things like analytical chemistry, which is fundamental to all of our degrees, is part of the curriculum, is that we're the powerhouse in terms of providing technical workforce people to our environment. Orange County, we have a lot of local students. But we have a lot that also go on to the best kinds of institutions all over the country. So, a lot go to PhD programs, including places like Harvard and MIT and Stanford and Berkeley and et cetera. And some of them go into science teaching, master's students, for example. And they go into pharmacy, and they go into veterinary medicine, and they go into MDs, of course, MD PhDs in part.
Great.
Yeah.
So what advice would you give to a student starting out in a career in science today?
Yes. Well, I would say go for what you love. If you get interested in science, and you really are interested in understanding how knowledge comes about and want to contribute to that, don't let all the people who are telling you that there are no jobs out there get in your way. I mean, we really, when you look at our graduate programs around the country, we have an awful lot of foreign students. And why can't we replace them with some of our own students? We really need to try to have that happen. And science training in itself, even if you end up in something else, is very valuable. I think what we learn in becoming scientists can be applied to many other kinds of jobs. And so there's a lot of possibility if you get a science degree, even if you don't end up going for a PhD or working in industry and so on.
Maria, you've worked with iron and you've worked with copper. For which of those metals do you have the highest affinity?
Oh, gosh. It's really hard to say because I love both of them very much. Right now, I've been focusing on copper much more in copper-iron interactions, but I really love iron as well, and there's a lot of work still to be done there that I look forward to maybe contributing to a little bit.
Thank you. Thank you, Maria, for telling us more about your career and helping us appreciate your career path.
And thank you, Bryan, for being willing to interview me and doing this so nicely.
My pleasure.